today we will finish the first chapter of biotechnology, which is principles and processes, and probably also start some applications of it because they both are connected. So we'll just take them in one go. So in the last class, I think we finished with cloning vector, gel agarose electrophoresis, agarose gel electrophoresis, and restriction enzymes. I think this is where we started, right? Okay. So anyone would like to give a quick summary of yesterday's class? So Varshini, this is something that you might also have to do. So in every class, the previous class is summarized in 5 to 10 minutes. And then we start with something new, okay? So since this is your first class, so you can just see and learn, and one day, like one day means probably tomorrow only, it can be you or anytime. If you continue with the classes, okay?
Okay.
Yeah, great. And there are two things that you should remember in my class. One is you don't have to take anything as a like a word set in stone just by face value of it. Just because the teacher is teaching things to you does not mean that it's unquestionable. You have all the rights. Nothing in science makes sense, if it is not in the light of evolution or proof. So you can ask, clarify doubts. If I'm teaching you something, don't just memorize, because it becomes very difficult. Biology is a very vast subject. If you just keep on remembering and memorizing things, it's impossible to understand biology. But if you understand the phenomena, it becomes easier, okay? So ask for proofs, contribute, discuss. Even you can, if you don't agree with, or there is no logic in something, or you're not able to understand the logic, you have all the right to ask, okay? Ask any number of times, but do not just rely on your memory, okay?
Sure.
Yeah. Great. So anyone would like to give a quick summary? Fatima? Can you hear me, Fatima? I don't think Fatima is able to hear me. Okay, so anyone who can hear me, Hiba, and anyone from 3, Atif, Donna, Hiba. Anyone would like to summarize, please? Please step up. No one wants to give a summary? Donna? Okay. So if no one wants to give a summary, then there'll be no summary. Let's begin. See, someone has to give a summary, because the class can be just as interesting as you make it, right, people? All of you have NCERT with you right now, your books?
Yes, sir.
Yeah. Okay. So please open your NCERT and we'll come to the final part where we are putting everything in pieces. Please come to page number, I don't know which page number is this. It's 201. Please come to page number 201, where it's the processes of recombinant DNA technology. So mostly all of these steps we have studied. So I'll just summarize it for you. So in order to make or process a recombinant DNA, or to make a recombinant DNA and get it expressed into another host system and get the gene product, which is the protein, we have to go through the following steps. So we first have to isolate the genetic material, which simply means that whichever gene we are interested in, it must be present in some or the other organism. Now it could be a plant, yeast, animal cell, any cell. So we have to isolate that genetic material, which is the DNA, from that cell. Now, since we cannot just isolate the gene that we are interested in, we have to take out all the DNA. And once we get, so yesterday in last class, we discussed what are the important things required for the isolation of any genetic material. I'm just summarizing this thing. So what are the steps that we need to isolate DNA from any source? So we need to take the cell, and in order to reach the DNA, we have to break open the cell, right? We have to break open this. By this, I mean that the membranes, there are membranes have to be degraded. Because unless and until we degrade the membrane, both the cell membrane and the nuclear membrane, we cannot reach to the DNA. So to break open the cells, we use enzymes. Okay. And these enzymes could be, let's say, lysozymes. If you are trying to break open a bacterial cell, for that matter, so use lysozymes. If you are trying to break open a plant cell, then you would need cellulase as an enzyme, because plant cell has a cell wall which is made up of cellulose. It's a cellulosic cell wall. So for plants and algae, you need cellulase. If you're trying to get out the DNA from a fungi or from yeast, which is a unicellular fungi, you will need chitinase, because they have a cell wall which is made up of chitin. So you need chitinase. So these are basically enzymes to break open the cell, right? In your schools, like yesterday we were discussing, or sorry, in the last class we were discussing that in schools, when DNA isolation is done as a part of the practicals, we don't use any of these because they are expensive chemicals. So there is an alternative, which is a cheap, handy, easily available alternative, which is detergent, right? You can use detergent because detergent breaks open. It works, but then detergent is not specific. It is harsh on cells, and it can also be used. Now, once we have opened the cell, after opening, what do we need to do? We need to get rid of the contaminants, right? What could be the contaminants in the cell? Remember, we just need the DNA. And what is DNA? What is DNA as a molecule?
The genetic material, like.
Yes, it's a genetic material. I've written here, isolate the genetic material, which is DNA. But what is DNA? It's a protein, it's a lipid. What is its chemical signature? That's what I'm asking.
It's a nucleic acid.
Yes, it's a nucleic acid. It's a kind of nucleic acid. There are two types of nucleic acids, very similar to, not very much different, but still they have a difference. One is RNA and one is DNA. RNA is ribonucleic acid. DNA is deoxyribonucleic acid. And just because it's deoxy, it's double stranded, and it is less reactive and more stable than RNA. So when I say get rid of contaminants, I mean we do not need proteins, because they are also present abundantly in the cell. So when you will break open a cell, you will not just get DNA, but you will get a lot of proteins, lipids, sugars, right? And also RNAs. So we want to get rid of all of this because we don't need it. Yes, we can use detergent mostly for any kind of a cell. It's just like killing the cell, simply. Okay. Yep. Cool. So we want to get rid of it. So to get rid of it, we again use enzymes. Now these are degrading enzymes. So for this step, the enzymes that are used for proteins, we use proteases, right? For lipids, we use lipases. For sugars, you can use amylases. So they will all be degraded. And for RNA, you can use RNases. Okay. We will not use a general nuclease because that will also cleave or cut DNA. Just specific nuclease, which is RNase, that will only degrade anything which is single-stranded nucleic acid, which is RNA only. So all the DNA will be protected. So ultimately, this is important in laboratory conditions. But again, when schools, we do DNA isolation for the sake for experimental purposes to teach you. They don't use any of these enzymes because again, they are very expensive. And so the kind of DNA that you extract in schools might have a lot of contamination. Okay. After this, so first is this treatment, second is this treatment, and then third is we need to precipitate out the DNA, because that's the only thing which is left now. Now to precipitate out DNA, we use chilled ethanol. So if you add chilled ethanol very slowly without agitating the tube, what happens is DNA molecules, come out in the chilled ethanol layer. They precipitate in the chilled ethanol layer. And that precipitate is all your DNA strands precipitated together. And you can spool it out as multiple DNA threads. In schools, remember again that it might have a lot of contamination, but when we want to do it for biotechnological purposes in labs or like serious research, we need to follow each and every step. Okay, so chilled ethanol based extraction of DNA is done. Once you get the DNA, now what will you do? So this is isolation. We did the isolation. What after isolation?
Cut the DNA.
Yes, perfect. After isolation, we need to cut the DNA because we don't need the whole DNA. We don't have any work with the whole DNA. We are just interested in any particular gene whose protein product we want to make through a DNA recombinant technology. We want to generate on a large scale. So we have to cut the DNA using restriction enzymes, right? Specifically restriction endonucleases. Now restriction endonucleases we have already discussed, and whatever restriction endonuclease we will select based on where we want to cut on the DNA, we will use the same restriction endonuclease for our plasmid vector as well. So both have to be cut with the same. So cutting is clear. After cutting, what do we need to do? There will be many fragments generated of the DNA, not just, so we will not to know, but we will know that what is the gene size, right? So that we have already discussed yesterday, that through agarose gel electrophoresis, we can separate out the DNA of interest, correct? So third step is separation. We call this process elution as well. We can elute out DNA from that gel. Separation and elution of DNA or gene of interest. Okay. Once we have separated it out, at this step also you can make or you can prepare recombinant DNA. But one more thing that is done in research or in labs is amplification of the gene of interest. So we want to make millions and billions of copies of our gene of interest so that it can be, basically the idea is to do it as a high throughput technique. we can do it on a very large scale, because we are not just doing it for an experiment, we are doing it for making a product that can be prepared and sold by these companies for as drugs or like medicines for certain things. So fourth step is amplification of gene. Now this is what we are going to do today. First thing, in a little detail. So amplification of gene of interest. Using PCR. So anyone knows what is a PCR? Anyone has heard this name before in schools or while studying? You must have heard RT-PCR a lot in last 2-3 years, right?
Yes,
Yes,
sir.
Where have you heard RT-PCR?
for the COVID testing.
For the COVID testing, right?
Yes.
Yeah. So what we do in that, any idea? How do we test for the COVID?
Like when I got it,
Mhm.
they put like a cotton bud in your nose and they take it.
So what are they doing by this?
Sorry.
What are they doing? Why are they putting a swab in your nose?
So they could take to see like if the virus is there, right?
To take the sample, simple. You
Yeah.
just want to take a sample of your internal nasal fluid in the mucus, and what are they going to do next in that sample? How will they know in the sample that there is COVID virus or not? You cannot see virus, right? Viruses are not like bacteria that you just put it under the microscope and you can see them. Viruses are very tiny and you cannot see it under the microscope. So how are you going to figure out that whether we have a viral infection or not, whether we have a COVID infection or not? Any idea? The idea is that you know that they are doing RT-PCR with your sample. So what is RT-PCR?
Maybe you need to check for the viral DNA.
Viral DNA. Check what? How will you check for a viral DNA? Testing for antigens. Antigen antibody testing is different. That's a rapid testing that you get done. So we don't test for, we can only test for antibodies or antigens that, antibodies that have been prepared in our body against COVID, but that can be done post-infection. So if you get your antibody testing done and you do not have any antibodies, which means you are not vaccinated and you also have never got COVID till now. But if you get antibodies, that does not mean that you are currently infected by COVID, you have COVID. It simply means that you were at least once infected by COVID and you have antibodies against it, which we can detect. So that's the rapid antigen test. That's different. That result comes in like just quickly. But what are they doing in PCR? Atif gave a very close reply, not true, not exactly the answer though, but testing for DNA. Yes, so we know that COVID is a virus and COVID is a novel virus. So there must be something which is only present in COVID, right? A characteristic feature of COVID, some spike protein and a gene for some spike protein, or any gene which is COVID specific. So we will just target that gene and we will amplify that gene. Amplification using PCR. So in reversed RT-PCR, COVID is a RNA virus. Its genome is RNA. So we first have to convert RNA into DNA and then amplify that DNA. Okay, that's called reverse transcription PCR, RT-PCR. Do you understand, everyone? In case of RNA viruses,
sir.
we have to do RT-PCR. If you are doing it for a DNA virus, you can just do PCR because it's already a DNA and you have to look for a gene, particular gene in this. The same thing, and to look for it means the number will be very less in that sample in your nasal swab because only a few particles, or depending on how much infected you are, you will get particles. So when we amplify it using PCR, we also get to know what was the viral load to begin with. If there was one virus to begin with, 1 will become 2 in the next cycle. When we are amplifying it, it will become 4, 8, 16, 32, 64. So to get to 64 cycle, it will take 6, 7 cycles, right? To get 64 in number, let's say. But if you are heavily infected, if your swab already has 8 viral particles, then to go to 64, you just need 3 cycles, right? 16, 32, and you reach 64. So in how many cycles you reach that detectable limit will tell you how much severe was the infection. Do you understand, everyone? So how PCR works is, it's known as polymerase chain reaction. Polymerase is the enzyme, DNA polymerase, chain reaction. Chain reaction means it's a chain. We are doing a series of reactions one after the other to amplify any region of DNA. We can do it for COVID. We can also do it here. So the DNA that we have cut and then separated through electrophoresis. Now we need to amplify it. So how it works is, let's say we have a DNA, a double-stranded DNA. Okay. So you know that DNA is like this, correct? Now let's say that from this base to this base is our gene of interest. Okay, this is what we want to amplify. So first is, if it is a double-stranded DNA, it will have 5' to 3' end, and the other will be 3' to 5' in the same direction. So this I'm referring to as dsDNA, double-stranded DNA. Why I'm saying double-stranded DNA? DNA is double-stranded only, but in this context, the next thing that we have to do, remember how was DNA replicated? When I taught you DNA replication in genetics chapter, how was DNA replicated? What was happening inside the cell? We have to just make the same steps happen here artificially, and the DNA will replicate. So if I have a double-stranded DNA, and to replicate it, first thing that I need to do, what do I need to do?
Need to introduce the enzyme for DNA replication.
But how will the DNA replicate? The DNA is stable, bound. First, you have to open the 2 strands. Forgot? If you cannot open the 2 strands by using enzymes, like there is helicase which unwinds, then there is topoisomerase, and all these enzymes I have taught you about. They have to open the 2 strands and make a replication fork. So people are not going back and not revising at all. So that's not a good sign, but it's your call. There is only till a point I can do as a teacher. So DNA first have to be opened. Now, inside a cell, there is a whole molecular machinery, because it's happening inside a cell at the will of, I should not use the word will, but in a synchronized process that cell undergoes. So there is a whole bunch of machinery that is recruited. But here we just want to amplify this region. We don't worry about the whole cell. So anyways, this piece of DNA we got. So whenever we have to amplify a gene, we cut slightly more. So these are called flanking regions. This is the only region we are interested in. This is our gene, but we will have some flanking regions on both the sides. Now, a very easy way to open a DNA is that DNA is heat labile. If you heat a DNA, DNA gets denatured. Denaturation means DNA opens up. Okay, so this is the first step called denaturation. So you take a DNA, double stranded DNA, and you heat it. And by heating, I mean something around 94 to 95 degrees Celsius. If you heat it, DNA get denatured. Okay, we don't have to boil the DNA. Okay, and the DNA is in a solution, so we are not going beyond 100, just 95 will be enough. Now, once you do that, what happens? The two strands open up, correct? This was the 5' to 3' strand with its bases. Remember, DNA bases are connected to each other by hydrogen bonding. All these lines I'm making, they are adenine, thymine, guanine, and cytosine, correct? ATGC, something like that.
Yes, sir.
So it will open, and again, I will mark the region that we are interested in. Let's say this is the region that we are interested in. So the same thing here. Now we have to just replicate this one. So what we will do is, we will design some primers. Now remember, DNA replication has a direction. Anyone remembers in which direction DNA replicates?
5' to 3'.
5' to 3'. Perfect. So DNA replication for this strand, there is the half strand is already there. The half strand will be replicated. This is called semi-conservative method of replication. So replication will happen in this direction, correct? So for these few bases, we will make a primer, which will go and bind like this. Okay.
Yes.
And for these strands, this also will make primers. Here, the direction of synthesis will be this. Now it will go in this direction, it will go in this direction. Once, so these two are primers. So one is called forward primer, one is called reverse primer. We have to put both the primers. If you just put one primer, then DNA, both the strands will not be replicated. Only one strand will replicate. So when we put these primers, so from denaturation to this step, when we are annealing primers, so this step is also just known as annealing, because we are annealing, we are ligating the primers. So from there, DNA replication can begin. But just by putting primers, replication won't start, no? We have to do something else. What do we need to do? If I just put primers, what causes DNA replication? There's a very important enzyme that you all have studied.
DNA polymerase.
Perfect. I also have to put DNA polymerase. Okay. Now for annealing, the temperature is around 72 degrees Celsius, but it's still very hot. Do you think DNA polymerase will be functional at this high temperature? And if we bring down the temperature, annealing will not happen properly, and these two strands will bind again. So we need a DNA polymerase which is stable at very high temperatures also. Now, human DNA polymerase or any other animal DNA polymerase that live around us will not work clearly. So we have to find an organism that normally lives in very hot environment and still does replication and everything. Any idea? Any candidates? Can you think of any genera or any kind of organisms that live in very extremely hot environments? Can't keep forgetting things like this. Which is the group of organisms that can be found even in high salt conditions, high acidic conditions, very hot temperatures, very cold temperatures?
Bacteria.
Bacteria, perfect. They're called extremophile bacteria, remember? And one of the extremophiles are thermophiles. So thermophilous bacteria, so scientists know that there are some bacteria which are thermophilic. They live in very hot geysers, but they live well. And if they're living well, they must be replicating their DNA. They also must be dividing and doing all sorts of things very, very easily. So how are they able to do DNA replication at very high temperatures, living in that hot boil, like almost boiling water? So they realized one such bacteria is Thermus aquaticus. Thermus means heat loving and aquaticus means it lives in, it's aquatic. So this bacteria has a polymerase which is known as TAQ polymerase, Thermus aquaticus polymerase. And this polymerase is thermostable polymerase. It works and replicates the DNA even at high temperature. So we will use this polymerase plus what we need to give to make DNA. What is the monomers of DNA? Nucleotides. So we have to give deoxynucleotides. These are synthesized artificially and we can easily synthesize a nucleotide. Now going back to the basics, you all know what is a nucleotide, right? It has three components, a phosphate and a sugar backbone and a nitrogenous base protruding inside from it, right? So that one thing containing these three components is called a nucleotide. That's why DNA is known as polynucleotide. So we will use these deoxynucleotides in the presence of polymerase, and what it will do is that, so these were the already existing strands. Once again, 5' to 3', 3' to 5' with their bases protruding inside. Remember, this is the region that we are interested in, let's say. And what we have already put the primer, right? So DNA polymerase will just keep making new DNA, right? We'll keep adding deoxynucleotides. You understand? Now this strand which is synthesized is 3' to 5'. Correct?
Yes, sir.
Or in this direction, 5' to 3'. Similarly, here also, there will be a new strand made due to tag polymerase, which will be 5' to 3'. Now we get this region amplified. So this part where we add tag polymerase is called extension, because we are extending the newly synthesized strand. So these three things are in a cycle: denaturation, open up the DNA; annealing, anneal a primer, put tag polymerase and nucleotides, and extend. Now you have to do the same process multiple times. Again, these two will act like parent strand, and again after extension we will come back to this process. So one cycle is approximately around let's say 3 to 4 minutes, not more than that. For, let's say, for 1 minute you denature, then you anneal for 45 seconds or 1 more minute, then you allow it to extend for a couple of minutes and you're done. Okay, then again go to denaturation. So these two strands will again open, again primers will bind, again DNA polymerase and deoxynucleotides will extend. So in one cycle, one DNA becomes 2. So in the next cycle, it will be 4, 6, 8, 16, 32, like that. So if you do this for 30 cycles, let's say, this is the power of PCR. In just 30 cycles, you can get around 1 billion amplified gene. Amplified gene or DNA molecules, I should use. Okay. Now this is amazing, right?
Yes.
Now let's say this cycle, and looking at the cycle, if you start with 1, this is the case. But if I already start with 1000, if I start with 1000 in the first cycle, which might be the case when which might be the number of such genes in your swab. Then in the next cycle, in cycle 2, how many DNA fragments will I have for the same gene? If I start with 1000, in cycle 2, I will have 2000, right?
Yes, sir.
Similarly. So it doubles every cycle. So if it is 1 billion at 30th cycle, can anyone tell me at 29th cycle, how many DNA amplified strands were there? This is not a difficult question at all. It's very easy mathematics. If after 30 cycles, you get 1 billion DNA molecules, how many will be at 29th? Donna says 9 million, and this is where you do the mistake. You will get half billion, 0.5 billion, okay, which is 500 million. 1000 million is 1 billion. So at 29th cycle, you will have 500 million DNA molecules. And at 28th, you will have 250 million DNA molecules, because in every cycle it is doubling. Remember that. It's the same question that they sometimes ask, that is it clear, everyone?
Yes,
Yeah.
sir.
Yeah. So if I'm culturing a bacterial colony, and in every 20 minutes the bacterial cell divides, and I start with 1/4 glass of the bacteria at time 0. Okay, so after how many minutes the glass will be completely full?
Sir, can you repeat the question?
The question is, a bacteria takes 20 minutes to divide. I'm culturing the bacteria. I started the culturing with a bacterial cell population which fills 1/4 of a glass. This is time 0. I have 1/4 of the glass filled with bacteria at time 0. At what time? How many minutes? After how many minutes the glass will be full?
Sir, 40 minutes?
40 minutes, right? Perfect. Cool. So this is called PCR, polymerase chain reaction. Each cycle has 3 steps, and you do approximately, you can actually do it any number of cycles you see fit. By this we can get millions and billions of copies of our gene. This is kind of a gene cloning only. And after this, what we need to do, we need to make vector, right? So 5th step is to prepare recombinant DNA vector or plasmid, right?
Yes,
So
sir.
if we have 1 billion amplified DNA molecules, we can use 1 billion plasmids also and make 1 billion DNA vectors, recombinant DNA vectors. And to do this, we use enzymes like ligases, right? Ligases ligate our DNA of interest to the plasmid vector, which is circular. And once we get, so this step is also known as, it helps in insertion. Once we make these recombinant DNA, then we would like to insert. So insertion of recombinant DNA into host. Now, what is host here? Host cell or organism. What is the host here? Host is where we want our gene to be amplified so that we can extract it. And what will you use as host?
Bacteria.
Yes. So host is bacteria. In some cases, it can be yeast also, which is because it's a unicellular unicellular eukaryote, and it's a unicellular prokaryote. So if it can work in bacteria, you use bacteria. If something is complex and some genes only are eukaryote specific and you're working with those genes, then you take yeast also according to your. I told you while teaching you DNA fingerprinting about two kind of vectors, BAC and YAC, right? Bacterial
Yes, sir.
artificial chromosome, yeast artificial chromosome. It's nothing but the plasmid itself. So here you understand how we make it, right? And remember, when we are inserting it, this BAC or YAC will also, this plasmid will also have a selectable marker about which we studied yesterday, right? That selectable marker helps us to select the transformant cells or the transformant bacteria and then culture them. So the next step will be to culture, simply to obtain the foreign gene product. I'm calling it a foreign gene because the host in which we have put this gene, it's not that host's gene. So it does not do any function in the host. It's a eukaryotic gene which I am expressing in a prokaryote. Let's say I want to express insulin gene and get the insulin protein. So I am just expressing an insulin gene into bacteria. So that's why it's a foreign gene product. So how to obtain it? To obtain it, we will use big containers. Okay, so in industries, this is an industrial step where big containers called bioreactors are used. So bioreactors are large vessels which can hold large volumes up to like 1,000 liters or even more of culture, of bacterial culture. So basically you want to make these bacteria culture for days and weeks, where the number reaches into billions and billions of bacteria, each bacteria having your gene product, your recombinant gene, and producing protein inside the cell. So basically you are interested in the protein. So after you have multiplied and synthesized the protein, this part is called biosynthesis. Once it is done, the desired protein is produced in very high yields. Then we have to just do the protein purification by killing all these bacterial cells, bursting it, purify our protein, and protein purification again can be done. The principle is something similar to DNA purification. There you are using things to purify DNA and you are degrading everything else. Here you will degrade everything else, but you will just take your protein, or even if you don't degrade everything else, you will just pull out your protein of interest. And it has many ways to do it. That is beyond the part of beyond the scope of your syllabus currently, but just know that protein purification can be done. This is how we have purified all the proteins that you get, except also your whey proteins. You know, whey protein are the most common type of protein that people take as supplements. Bodybuilders or people who go to gym take whey proteins, right?
Yes, sir.
So it's a purified protein. It's not very good for the system in a long run if you are just taking whey protein and not exercising properly, or because protein in low protein deficiency can lead to problems, but protein excess in the body where it's not metabolizing properly can also lead to problems. Toxicity, it can lead to toxicity. Anything in our body which is more than what is required, either body degrades and throw it out, and if it cannot do that, it leads to toxicity. Even oxygen, something as vital as oxygen, we need oxygen in a particular amount, in a particular partial pressure. You cannot give 100% oxygen into the lungs. That will just kind of combust or burn your lungs. Okay, so bioreactors are large vessels where to culture bacteria. Okay, I'm calling it a transformant bacteria because it contains our gene of interest. It's a biotechnologically designed bacteria. And these bioreactors have controlled conditions. All the conditions you can control. What conditions? For example, pH, temperature, humidity, salt concentration, etc. Okay, vitamin, substrate, everything. So everything has to be controlled depending on what you want to do, even oxygen, CO2, etc. So once you have produced a large amount, and this tank, these bioreactors are constantly, you keep stirring it so that everything is in the reaction mixture. Oxygen is available to all the bacteria to live, survive, nutrition is there. So you make sure of that. Once you see that the number of bacteria has reached the maximum limit which you wanted, now you need to do downstreaming processing. Now, what do we mean by downstreaming processing? Simply, we need to purify the product, which is the protein, and make it ready to be to become a finished product or a marketable product. Now, in downstream processing, write down protein purification. You purify the protein. Okay. Then you put that protein in a formulation. Okay. What do I mean by formulation? We also have to like protect the protein. Proteins can degrade, right? A protease or anything, if I just put protein anywhere, it can degrade over time. So we have to make sure that this protein that we have purified after doing all this very, very long and cumbersome, painful process, a proper formulation is there. So formulation is how these proteins are converted into medicines. Okay, formulation with suitable preservatives. So every medicine comes with this formulation where it is stable, starting with something as common as paracetamol to any other protein injection, tablet. All medicines have to come with formulations where they are stable. Now, these formulations go for clinical trials. If it is a drug that you are preparing, it will go for clinical trials, or what do you call it? Quality control testing. If it is something which needs to be marketed, there will be quality control testing. Okay. And finally, the product is ready. So this is the overall process of recombinant DNA technology, starting with isolation of that DNA, getting that gene, separating it through gel electrophoresis, amplification using PCR, which I told you in detail, preparing a recombinant vector or plasmid, then inserting it into a host organism, culturing it in bioreactors to obtain the foreign gene product, which is protein, then downstreaming processing, purifying it from the bacterial cell, making it into a formulation and sending it for whatever use we are creating it for. Is that clear, everyone?
Yes.
Yes, sir.
So as we started with, in summary, you realize now that biotechnology, the whole field or the industry, deals with a large scale production and marketing of products and services or processes using live organisms, cells or enzymes or proteins and their components, right? This is what we began with when we started the chapter, this was the definition with which we started, right? And the key process, I told you there are two key processes in biotechnology. One is genetic engineering, which is designing a DNA, recombinant DNA. Okay. And the second is process engineering. So this whole process that we have designed, that is also biotechnology. That first we will do this, then we will isolate, then we will use restriction enzymes, then we will cut the DNA, we'll cut the plasmid using the same restriction enzymes, sticky ends will be produced, we will ligate using ligase enzyme. So all this is process engineering. So is the whole chapter clear to everyone?
Yes, sir.
Yep. Okay, great. So I'll stop here for questions. I'll stop here for a couple of minutes for questions. If you have any questions and doubts, please go through all your notes, and I give you a couple of minutes here. And then I will start with applications, the chapter applications of biotechnology, the next chapter basically, where we will, I'll tell you about the fundamentals again. So we have to study applications in agriculture, medicine, and making transgenic animals. These three applications are in your syllabus. So since you have understood the whole process, it will not be very difficult to move forward. But if you have any doubts about anything, any process, any step, any tool component, please ask right now. So I give you a couple of minutes, please go through all the notes, and then we'll begin with the new chapter. Okay, so any doubts, any questions?
Sir, can you explain selectable markers?
Yes, sure. So let me go back. Where did we study selectable markers? Yes, here. So as I told you, when we are designing this whole plasmid thing, right? Our recombinant DNA, which will contain our DNA of interest, right?
Yes,
And
sir.
then we will send it inside the host. And for that, I told you that we have to make the host, which is the bacteria, competent, right? So that it can take up our plasmid from the surrounding. Now, there are ways to do it. One is you can directly use microinjection and put it inside the cell. But by that process, we can just, if we manually microinject things in bacteria, how many bacteria do you think you can inject? Limited, right? In a day?
Yes, sir, not allowed.
Yeah, not at all. It's not useful. Then there's something called gene gun. You just put your DNA of interest, coat it over tungsten and gold microparticles and bombard the cell with it. So these will enter inside and also take our recombinant DNA with them. Third way is to make them competed through heat shock, right? Keep them on ice, let the bacteria be on ice, then suddenly take them to 42 degrees, which is, a heat shock. So the pores of the bacterial cell wall will open and that will give enough time for our plasmid to enter inside. But by any of these methods, we cannot be sure that all the cells will get our plasmid for sure, right?
Yes, sir.
So how will you know when you're culturing the bacteria? Let's say you started with 100 bacteria and you want our gene of interest, which is in the form of a recombinant DNA. We did this processes of making competent or gene gun or let's say heat shock. But out of that 100, we are not sure how many actually got transformed, right? How many are now the transformants carrying our gene? If they are 50, 60, 70, and we would not like to keep culturing all the 100, because if I put all the 100 in that bioreactor and they become, let's say, 10 billion, then out of that 10 billion, only 6 billion might be the transformants, right? Then it's a waste. Yes or no?
Yes, sir.
So if I can early on somehow only select the transformants, how can I do that? For that, I use a selectable marker. So this selectable marker could be an antibiotic resistance gene. So what I will do is, along with my gene of interest, I will also insert a gene which will give resistance towards an antibiotic in that plasmid when I'm sending that plasmid inside the host. Now, I just have to check which all are transformants. Remember, whoever is transformant will have antibiotic resistance gene. So I will just, before culturing it into bioreactors, I will put all these bacteria into culture plates carrying that antibiotic. So those who will not have my plasmid inserted in them will die, right? Because they don't have any antibiotic resistance. Correct? Because antibiotic resistance will come through the antibiotic resistant gene, which is there in my plasmid, which I created, the recombinant DNA. Yes or no?
Yes, sir.
So antibiotic resistance gene here is working as a selectable marker for me. So that way, all the non-transformants will get killed and only the transformants will survive. Now I can take all that transformants, whatever number they are, even if they are 10 from 100, I know that these 10 are for sure transformants. And when I will culture these 10 to make 1 billion, all the 1 billion cells are transformants. I will get good yield of the protein at the end. Correct?
Yes, sir.
So that's selectable marker. Is it clear?
Yes, sir. It's clear.
Okay. Any other question? Anyone? Okay. So if there are no further questions, then let me start a different chapter, chapter 12, which is what it is named, biotechnology and its applications, right?
Yes, sir.
Okay, great. Now, since you know the processes and the fundamentals, applications is not going to be. No. What are the fields where you think this can be applied? It can be applied to a multiple, almost everything you see around, which is a product coming from a living system, starting from organic cosmetics to your shampoos, to drugs, to medicines, to food that we eat, all are product of biotechnology these days. But we will primarily focus on the two things. One is the field of first important thing is food. So food production, how biotechnology has been used for food production, or I can just say agriculture, right? And medicine. Okay, or the health sector. So these two are heavily influenced by biotechnological applications. Now remember again, can anyone just summarize or just briefly tell me what was the objective of biotechnology? What is the objective with which we started? By now you should be able to answer at least the objective. What does biotechnology do? What is the definition of biotechnology?
To integrate living systems with techniques to produce services.
To produce either good products or services for humankind, to improve the quality of human life, right? By providing better health services or by providing better nutrition is the are two most crucial ways to improve the quality of a human life, correct?
Yes, sir.
Now, when I say that biotechnological applications in agriculture, let's start with this part here. So when I say that I am using biotechnology or I am doing a biotechnological application in agriculture, what do you think I'm doing or scientists are doing? What do we mean by this? What we want to achieve? What is the existing problem with agriculture that we want to solve by biotechnological applications or biotechnology? Basically, why are we using it for agriculture? What is the problem?
To increase the yield, like to produce better crops.
Exactly. The only problem is growing population. If anything that is happening to planet Earth or happening to humans, starting with something as common as food, to waste management, to ecosystem, to extinction of organism, to global warming, to bad economy, finances, poverty, anything, if you just trace it back enough, it will be linked to overpopulation of humans. We are way beyond our capability, like to provide everyone with good resources, food, water, etc. So the only thing which we have to focus and work on with respect to agriculture is produce more from the limited field that we have, right? In the beginning, when we did not have biotechnology, or the field was not developed, people started expanding. As human population expanded, they started expanding their agricultural fields. We started clearing jungles and making them agricultural fields, right? And even cities, correct? So all our cities, towns, villages, agricultural fields, they were all forests and jungles before. We reached to a limit where we realized that now if we go any further, we will be creating disbalance in this ecosystem, because then we are invading the territory of these animals. And COVID is the recent example of what happens when humans keep invading animal territories. The viruses that were not infecting humans, it's not just then that when we are invading their territories, we are living in close contacts for very long, we are just causing harm to them. The harm will, it's like a boomerang, it will come back to us as well, because the more we intermingle, the more our viruses will also, our bacteria, our pathogens will also intermingle, right? Yes or no?
Yes.
Correct. So if you are making your villages, cities, and fields in areas where bats live, correct? So where will bats go? Bats will just live there, but their fecal matter, their urine, they will eat fruits. So their half-eaten fruits and contamination in that will somewhere come back to us and interact with human system, right? So we will try to kill them, smuggle them, eat them, things like that will happen. And the more there is cross species interaction, the more is the interaction between their pathogens as well. And the more those pathogens get a chance to mutate, because they are anyways mutating in nature every time, every now and then. Bacteria and viruses are very fast mutators. They evolve faster than higher life forms. And this we have seen that in just last 3 years, how many strains of COVID have come up? Some are more lethal, but some are more transmissible, less lethal. So you must have heard names like alpha, beta, gamma, delta, omicron, epsilon, this kappa, this and that, right?
Yes, sir.
So they're all variants. They're all slightly mutated, different variants of the virus. So this is what happens. Now, after realizing that, the next quest is, let's see, we cannot keep expanding and keep destroying every ecosystem and keep making everything as a agricultural field to grow more crops for the growing population. So what else we can do? With the same limited amount of land, we can increase the yield. Now, how can we do that? Now, to increase food production with the same given amount of land, we have to resort back to scientific technologies. One is biotechnology. So the whole motive is to increase food production, or to make high calorific or high nutritional value food. We all know about rice, correct?
Yes, sir.
What is rice a major source of? What does rice provides us?
Carbohydrate.
Carbohydrate. They were supposed to provide us carbohydrate. But now we have genetically modified rice varieties that provide us carbohydrate, proteins, and vitamins as well. Over time, so these are called genetically modified varieties or high yielding varieties, HYV, high yielding varieties of rice, wheat, and any other cereal, because we realized if we just grow rice as a source of carbohydrate, we also have to grow something for the source of vitamin. We also have to grow something for the source of protein, and growing all of this is not possible on limited land. So let's grow crops. So rice, for that matter, did not have in nature, in wild, they do not have genes, like genes inside them to produce vitamins or proteins much. So we introduced that gene inside rice. Okay, so that now in that seed of rice, which we will cook and eat, it's not just carbohydrate, there is also some vitamin and protein. Do you understand, everyone?
Yes, sir.
Yeah. So to increase food production, there are a few ways to do this. What are the ways? How can you increase food production? One way is you make chemicals which are called agrochemicals biotechnologically, and we do agrochemical based agriculture. Or agrochemical based farming to increase the yield. Second is what you can do. You can go for organic farming or organic agriculture. This is something you must have. It's a very, very used marketing gimmick word. These days, whatever you buy, they put a name organic in front of it, right? Even in front of things which are very obvious that they have to be organic. So you will see organic fruits, organic vegetables. What does it mean?
Use of chemicals.
That you are not using chemicals, right? Then how are you growing them? If you're not using chemicals, how are you growing them? Is the question, right? So you have to ask, is it true that are they really organic? Or it's like organic apples. So you will get a normal apple, let's say 50 rupees per kg in the sabzi market. You go for organic apples, which is like 500 rupees, you get 6 apples. Very beautifully, packed in plastic with a label and a sticker on it. But what difference does it make? Is it really organic? If it is organic, and how they have grown it then? Then third option is genetically engineered. You use genetically engineered crops. Okay, and you do a genetic engineered crop based farming or agriculture. Okay, this especially in recent times have taken up. People have started realizing that if you use too many chemicals, do chemical based farming, in a long run it's not very sustainable, and also the accumulative effects of these chemicals. Basically, these chemicals are fertilizers, and you know that what fertilizers provide, the most important vital elements required for a plant growth: nitrogen, phosphorus, potassium, right? Also, insecticides and pesticides are used as chemicals to keep these plants away from diseases. Then, so we give chemicals for their growth enhancement. We give chemicals to protect them from any disease, because we cannot afford our crops failures. We have to at any cost get yield. Organic agriculture, yes, it might be more healthier, but again, the point that scalability, if you are, where are you getting, it will take some time to establish organic agriculture everywhere, and it's not very, very fast. And the third is genetically engineered crops. And how many of you have heard of green revolution, people? Let's write it with a green. Green revolution that happened in India. Heard of it?
Yes.
What is green revolution? So there was something called white revolution. There was something called golden revolution also. And then there's something called green revolution. What is green revolution?
It was like a movement in India to increase the plant production.
It was a movement worldwide, but we study it in context to Indian population basically. But to do what?
To increase the yield and like the production of food in India from plants.
Food, which food, what food? And how did we manage to increase the production?
Improve the variety of the crops. The use of HYV seeds.
The both sentences mean the same thing. Variety of the crops and HYV. HYV is a variety of a crop, high yielding variety. The full form is high yielding variety. So we took high yielding varieties. What do we mean by high yielding varieties? How are they high yielding? These are the questions you have to ask yourself so that you are prepared, because they will be asked in entrance. If you wish to remember everything, then you better start doing this job and you do it good. But I just normally don't rely on my memory. So I rely on the logic, I rely on a story that I will remember, I rely on concepts. If any one of you think that memory is their strength, good if it is in reality, but to 95% of human on the planet, memory is not the best thing to rely on. We are not good with memories. We are not hard drives or computer chips. We are good with processing. Our brain has evolved to process, not to memorize. In fact, it's the latest research in neuroscience shows that our brain is designed or evolved to forget more than to remember. So focus more on concepts. That will be my standing advice for all kind of exams that you are planning to give in your life. So in green revolution, it was a revolution to increase the yield of every green thing, which is the food product. Plant-based food is comes under green revolution. So we managed to increase it to around 3 times, but it was not enough to feed the growing human population. And in the beginning, this green revolution was supported by better and better agrochemical practices or better management practices and better use of fertilizers and pesticides. So over time, they realized that the better fertilizers you keep making, their cost is also more. And the input that you are then putting into developing crops or growing crops goes up. So these crops, we increase the amount of crop, but it's also expensive. Okay. And it's the agrochemicals have a limit. So every farmer cannot buy these very expensive chemicals, spray it in their field and manage it effectively. So scientists, they brought a group of scientists together, botanists, geneticists. They sat and realized that let's look for genetical answers in the plant. Now, why do we use all these things, and what are the limiting points in agriculture? One is water, correct? Many crops fail due to no rains. So it failed to rain in that season and your crops will go bad, correct? That is one. Do you agree that is one limitation?
Yes,
crops need a lot more water to grow. Then why are we using chemicals? Because these crops are very susceptible to certain insects, which are pests, and to certain pathogens, which are bacteria, fungi. They easily get disease, right? So these are two things that they realized could be solved by genetics. So what they did, they started doing experiment that what if there are animals, right? So there are plants also adapted. For example, cactus. Cactus survive very well in very less water or drought conditions, right? In fact, you go to any region of the planet where you see life, you will see both plants and animals, some kind of plant or some kind of animal growing. So life grows everywhere, almost everywhere, except for Antarctica, because we don't know if there is any people have people, actually scientists hypothesize that there might be extremophile bacteria there, but I'm not sure how much we know about it. But apart from that, any habitable continent, we have life. We have either an animal. How did they adapt? So they realized there are many plants who can tolerate these stressors good. They can tolerate cold conditions. So even in countries which are cold, which do not have enough sunlight, these plants can grow. They can also tolerate drought. They can also tolerate salinity. They can also tolerate heat if cold. So they started manipulating their genes and playing with these to see if I tweak this gene, will the plant survive better in less drought condition, in less water. And by doing all that, and similarly for resistance against diseases, if there is antibiotic resistant gene in bacteria, why can't plants be resistant to certain bacteria and certain fungi? So by this, we created genetically modified crops whose genes have been altered so that they can survive better. Okay, but these genes were not just altered to make them survive better, but also to increase their final yield, because the final yield is the edible thing. For example, in a wheat plant, we do not eat the whole wheat. We don't eat leaves or the stalk or anything. We just eat that seed, the wheat, right? So if one plant is producing, let's say, 50 such structures containing, each structure containing 100 wheat seeds, can we increase it? Can we make these genes in multiple copies so that now on the same plant, instead of 50, there are 500 is too much stretch, but there are 100 now. I doubled it up, or 150 now. And each unit contains now not just 100 seeds, but 200 seeds. This way, in the same given land, I can make the plants produce more and also sustain better. Do you understand, everyone?
Yes, sir.
So that is one way using genetics. Okay. So I think we are out of time. So we'll continue this in the next class. We'll start with genetically modified organisms. Your homework is please go and read about genetically modified organisms and read a little bit about BT cotton, some processes regarding that from NCERT. And we'll do it in the next class. Okay, everyone? Anyone has any question? Any immediate question to ask? Okay, so if there is no question, I declare the class here and I'll see you all in the next class. I hope, Varshini, did you understood